My current field of research is focused on the elucidation of neural circuits that drive a simple behavior. An example of such a simple behavior would be decision making based on a sensory input, let's say a tactile input or a visual input. To put this in a context, it's estimated that about 1,000 decisions are made per day per human. In order to elucidate neural circuits, there are two areas of research or areas of knowledge which you have to understand decision making on a mechanistic basis. One is you have to have a 3D reconstruction of the neural circuit that underlies decision making. And the second type of knowledge is you must know the pattern of electrical signals that is sweeping through the circuit before the decision is made and when the decision is made. You have a 3D coordinate system, and within the 3D coordinate system you can register hundreds or thousands of neurons so that they will represent the anatomy of a part of the cortex, let's say of a whole cortical area. That's one part. The other part is that this network of cells then has to be made alive by introducing electrical signals and watch the flow of signals through this network. And the aim is to be as exact and as closely as possible to what's happening in the real tissue. What we have found out as major findings is the simultaneous representation of a sensory stimulus in different cell types, specific cell types. And secondly, we found out that the major determinant of the activation of these cortical cells by the underlying activity in the thalamus is the synchronicity of the electrical signals in the thalamus. The challenges in circuit analysis can be summarized by a new word that has been invented or created over the past few years, and this is called connectomics. It means that to elucidate a neural circuit, you have to have a detailed map, an anatomical map of all the cells that are participating in a neuronal circuit. And there are various ways to do this. There is a light microscopic level, but there's also now at the electron microscopic level, which will in the future enable us to have a complete 3D model of a pathway, or in the somewhat more distant future of a whole cortical area or of the whole cortex. The task is to look at large ensembles, at the general behavior at cellular resolution or even molecular resolution. The second challenge is to find out which of the mechanisms have been described under very simplified conditions are applicable in the intact organ. And the last challenge will be to find out which of these mechanisms that are present in human tissue is compromised in pathological conditions. And there are many examples, for example, Parkinson's disease, or there are many challenges.